Good afternoon, and thank you for standing by. At this time, all participant lines are on a listen-only mode. After today's presentation, you will have the opportunity to ask questions, and you may do so over the phone by pressing star one at that time. Today's call is being recorded. If you have any objections, you may disconnect at this time. It is my pleasure to turn the call over to your host for today, Ms. Irene Ahir. Thank you, ma'am. You may begin.
Thank you. Hello, I'm Irene Ahir of CDRH's Office of Communication and Education. Welcome to the FDA's 41st in a series of virtual town hall meetings to help answer technical questions about the development and validation of tests for SARS-CoV-2 during the public health emergency. Today, Timothy Stenzel, Director of the Office of In Vitro Diagnostics and Radiological Health in the Office of Product Evaluation and Quality, and Toby Lowe, Associate Director of the Office of In Vitro Diagnostics and Radiological Health, both from CDRH, will provide a brief update. Following opening remarks, we will open the line for your questions related to the development and validation of tests for SARS-CoV-2. Please remember that during the town hall, we will not be able to respond to questions about specific submissions that might be under review. Now, I give you to Timothy.
Thanks, Irene, and hello, everyone, and welcome back to another week. It's been a busy week at the FDA, and I'm sure a busy week for all of you. And again, appreciate all that you do. I think I said this at the beginning of these town halls, but somehow it snapped back to calling me Dr. Stenzel. I am Tim. So when you call in, just refer to me as Tim. Thank you. So I did want to give an update. We continue to see an amazing number of submissions, which is great. Many hundreds on a monthly basis, basically. We are also really have ramped up our decisions. So we are currently, over the last week, averaging about 9 decisions, that's N-I-N-E, 9, a day. And some of these are, of course, new original authorizations. They pop up as a new authorization on our FDA website. Many of them, though, are also supplements and amendments to existing EUAs. These aren't so easily spotted when we make these decisions. But if you go to a given assay and you click on the plus sign for that on the website, you'll see all of the updates to their authorizations, which are the supplements and amendments. Also, when we consider a decision in our accounting when we issue and close out a pre-EUA feedback. And then, of course, there are negative decisions, and those we don't publicize typically, nor do we publicize, when we've closed out a pre-EUA submission and give feedback to the developer. So not all of our decisions are publicly seen easily or at all. So just want to kind of explain that in a little bit of detail. But we are working harder and faster than ever with more people, whereas in the beginning of the pandemic, we were making, when we had an application, making maybe 1 decision a day. We're now making 9 decisions a day. So anyways, let's get started then. Let's open it up for questions. And Toby and I look forward to hopefully being able to help you today. Thank you.
Thank you. To ask a question, please ensure your phone is not muted. Press star 1, and when prompted, clearly record your first and last name so I may introduce you. Again, to ask a question, press star 1. And please stand by for our first question. Our first question is from Kay Taylor. Your line is open.
Hi, Tim. My question regards antigen tests for home use, either by prescription or over the counter, that incorporates the use of a mobile phone app. And wanting to look to see if we kind of have the general thoughts of FDA's current thinking in our head. Per the non-lab template, we would establish a set of critical smartphone specifications that we would outline before we would do any clinical testing or usability testing. Our thinking was to perform a clinical study with just a single representative iPhone model based on what we've seen with some recent clearances, EUA authorizations, and assuming, such as maybe the iPhone XR, and assuming that clinical study provided acceptable performance. Our question is more along the lines, what's FDA's current thinking about, would that then allow other mobile phone models and operating systems that meet that initial set of critical smartphone specifications to be considered acceptable for use as alternative devices? Or is it kind of a stepwise approach that FDA wants manufacturers to take? As you know, there's a lot of smartphone operating versions as well as models out there.
So is this for reporting purposes only, or is this to capture and analyze an image of the device to make a positive or negative call?
It is both. So it would instruct the user through the collection and testing process. It would, capture an image and interpret the result, and then also report it to authorities as needed.
So for reporting purposes, it is not a requirement for an EUA authorization, at least the time of authorization, to have a reporting feature. However, in what you described, we want to make sure during the review that the smartphone that you use to capture and analyze an image for determining whether something's positive or negative is accurate and reliable. And so obviously different smartphones are going to have different cameras and different software. So it would be important for us to take a look at what the risks are for using something that's not used in a clinical study. So I can't promise you that we wouldn't ask for some additional data on additional models or manufacturers if you're going to allow them in your for your test. However, that may not always require necessarily that you do a clinical study, but you do some sort of comparison study between the different cell phone models. So that your question is a great and important one, and it's best answered specifically with one of our reviewers through our pre-EUA process. So I wanted to give you at least some flavor of what we've been thinking about and what's important that would help you formulate your questions to our reviewer.
All right. Thank you, Tim.
Our next question is from Greg Slovatkin. Your line is open.
Yes, thanks, Tim, for taking my call. As you're well aware, there's a big debate going on, PCR tests versus antigen-based tests. And this covers a gamut of populations being tested. I mean, it's everything from, the claims that one test is better than the other when it comes to asymptomatic patients that are being tested. It also gets into the areas of what's more accurate. And I guess my question is, what is the FDA's current thinking on PCR versus antigen tests and when they could be optimally used? I guess that's my first question. Second one, and this doesn't relate to either a platform in particular, but why is FDA still issuing EUAs that require a prescription for a test? When there's such a desperate need to get mass testing done.
Okay, so molecular versus antigen thinking and RX. So RX, let's start with that, is a mitigation on risk. So if patients don't have a clinician involved with interpreting the test, then we look at other mitigations to ensure accurate testing, and that the reading of the test and the interpretation of the test is also good in the hands of the consumer. So we've authorized OTC tests that are without prescription, and so we are clearly very open to it. But we do ask for additional validation work for that, and as long as developers are willing to do those extra steps, we're willing to give an over-the-counter claim to that assay. Regarding molecular versus antigen, obviously we support both from the FDA perspective. Our job is to take a look at the different technologies that can be brought to bear to help this pandemic and make sure that they're accurate and safe to use. And that's our main consideration. We're very supportive of all types of testing, whether it's molecular point of care, molecular central lab, or antigen point of care, antigen home, molecular OTC, antigen OTC. So we see that they're all able to assist in this pandemic. We know that in general, molecular assays are more sensitive than antigen assays, and there are therefore some trade-offs both ways. I mean, some people have said molecular assays are too sensitive, and I think, if you're looking at asymptomatic people and you don't know when they got infected, a molecular test might identify somebody, a week or two or more after they became infected, and maybe they're not as infectious or are not infectious any longer. Versus antigen tests, which usually require more intact virus and aren't as sensitive. Probably, the good ones are the ones I think that we've authorized are good ones. Are probably better able to tell, who are the patients that are positive and truly positive. Those are probably patients that you want to make sure that you take appropriate measures to make sure that they don't transmit that to somebody else. So the other thing is, for central lab testing, especially very high throughput, unless you have an instrument and we have authorized one central lab antigen test, and that's fairly high volume, but it's a lot easier to make a molecular test and do it in large volumes in a central lab. But obviously a central lab is a high complexity lab environment, and a point of care clinic, would, not be able to do a high complexity lab test, but they can do a waived or a deemed waived test. And so, and also at home, it would be a similar sort of easy to use. And it's a lot easier to make an antigen test point of care and amenable to home than a molecular for obvious reasons. The other advantage that antigen tests can have is when they're good and they can be manufactured reliably, which is a hard thing to do. They can be made in the millions of tests per month or more. I mean, you've seen the press reports from some of the manufacturers, upwards of 50 million and maybe going up to 100 million tests from a single manufacturer, single antigen test per month. And those are volumes that, are potentially orders of magnitude greater than what molecular manufacturers and what individual labs, central labs can test for. So we look at the strengths and weaknesses of all the technology and weigh them and look at the benefit risk and if they can help out, we're very supportive. But they're very, different uses, I think. Hopefully that answers the question. It was a good philosophical question. Thank you.
Yeah, I just one quick follow-up. I know you took some time there. I mean, FDA has said previously that there's a higher chance of false negatives for antigen tests versus molecular tests. I mean, that's still the guidance, the feeling of the agency, right?
Well, very clearly, antigen tests are more likely to have false negatives, especially even and important false negatives, especially in the first, day 0 to day 5 or day 0 to day 7. Performance is clearly lower in that period of time for symptomatic people than a real good central molecular test. So you're going to have negative results with an antigen test that will be on somebody who's symptomatic and has SARS, and you'd have to do a lot of hand waving to convince me that somebody in the first few days of symptomatic infection can't be infectious. And they're going to be falsely negative with sometimes with an antigen test. They're less likely to be false negative with a good central lab molecular test. So it's why our labeling for antigen tests all say the negatives are presumed negative rather than negative.
Thank you.
Mm-hmm.
And our next question is from Marianne Feichner. You may go ahead.
Good morning. My question is also about a smartphone application for interpretation of an at-home test. As we all know, smartphone manufacturers often push major updates or security updates to their operating systems. What's the agency's expectation around system validation or updating after major updates to a smartphone operating system?
I think it's just a wise thing to do for a test developer to monitor that. I mean, ideally, a test developer would have relationships with the manufacturers of phones or sellers of phones that they're using, so that they can know ahead of time when an update might be coming. But if suddenly an update is made and it changes the performance of your test, the FDA is going to want to know that right away. And if you're giving now inaccurate results out, then that would be of public health importance.
And also, I would note that one thing that we recommend is that you discuss this topic specifically with your review team when you have your EUA request in, because we would be interested in seeing your plans for addressing periodic operating system updates, since obviously everyone knows that happens. And depending on how robust your procedures are for handling those, we may be able to build in some change control into an EUA.
Thank you very much.
That's
very informative. Thank you.
That's to make it easier for you to make updates and not have to check in with us. Thanks.
Awesome. Thank you.
And our next question is from Mark Delvecchio. You may go ahead.
Good afternoon, Tim. Thank you for taking my call. My question is related to molecular multi-analyte tests. If a test developer wishes to seek a sample pooling claim for the SARS-CoV-2 target, would it be necessary to validate and obtain a pooling claim for the non-COVID influenza or RSV targets, or is there a mechanism to obtain a pooling claim just for the COVID target? For example, a labeling restriction or something of that sort. Thank you.
Hey, Mark. So I'm just going to repeat it because you were breaking up a little bit there in the beginning. So you have a molecular multi-analyte test, and you want to do pooling. And what validation requirements are there for the or recommendations are there for the non-COVID targets? That's a question I don't know the answer to definitively. As far as through labeling, can we say that the other tests aren't covered. I don't know. I'd want to check with our team. I'm not sure if we've gotten this question before. In pooling, would you mask the other results? And so you would just have the ability to pool and just test for SARS with your multi-analyte test? Mark,
I guess that's a possibility. Yeah, I presume that's a possibility as well.
Yeah, well, that would be an effective mitigation, is that if someone wants to pool, they're probably pooling mostly for SARS, not for the other diseases, right?
Right,
and
exactly.
if they would mask the other results, then you would need to validate, for the other analytes if you mask them.
That's helpful. Thanks. We could follow up for the specific assay as well. I appreciate the thoughts. Thank you.
You're welcome. Thanks.
And our next question is from Wenli Zhu. You may go ahead.
Hello. Yes, thank you for taking the call. So I have a question here. So for the molecular test, if a test is validated for NP swab, it can be used for anterior nasal swab as well. Is that true for antigen test as well? Say, if a manufacturer said that their kit is good for NP swab, can I use it for anterior nasal swab, or automatically I can do that, or as the manufacturer, when we do the validation, we have to include the anterior nasal swab as well, not only NP swab.
Yeah, to talk specifically about the antigen test, yes, we would ask to see patient data for anterior nasal swab. The antigen tests are less sensitive. They're more sensitive to differences in viral loads that might happen between the nasopharynx and the anterior nares. So it's important to understand that performance difference. Now, we're not requiring for an antigen test that they do a nasopharyngeal swab. In fact, I encourage most to try to validate and get the recommended sensitivity with anterior nares or, worst case, mid-turbinate swabs, because those are much better tolerated. They can be, if you validate it for self-collection, they can be self-collected. They're then amenable, if you have a good antigen test and it's easy to use, for a home use, either prescription or over the counter. So, whereas a consumer cannot perform nasopharyngeal swab on themselves, at least not safely.
Okay, good. Thank you so much.
You're welcome.
Okay. And our next question is from Anna Gabrielli. You may go ahead.
Hi. We're developing a breath test for screening COVID. Last week I asked about the PPA or sensitivity requirements. You mentioned that you would think about it and discuss it and give an answer. I was wondering if you have any thoughts on the subject.
We do. I can give you some of our current thinking and philosophy as it comes to breath tests. This is all still being developed, so I would, but I think our team can provide you with even more specifics if you send an email to the template's email box. But I want to point you in sort of the flavor of where we're going here, so you understand and can ask good questions, hopefully. One is, when you think about the benefit risk of a breath test in aiding in this pandemic, if somebody is symptomatic versus somebody who is asymptomatic, the benefit risk favors testing the asymptomatic populations with a novel device such as this, especially if it has a high sensitivity in the asymptomatic population. Someone's symptomatic and it's a totally novel device when they're, when the concern about finding positive people is more moving into the asymptomatic population now as we try to control the spread of disease. Someone's symptomatic, they should be isolating, and masking and all those things, and get tests. But if the tests don't make sense, then. So the benefit risk really favors using a breath test in the asymptomatic population. We would want to see that, it has a relatively high sensitivity in the asymptomatic population. And since this is a novel technology and we don't have the same sort of experience, even with other respiratory viruses like we do with molecular and antigen, we will probably want to see more positives in the asymptomatic population than we would ask for an antigen or a molecular test for authorization of asymptomatic screening, just because we want to make sure. It's not going to be, orders of magnitude more. It's just going to be a little bit more, but we think still reasonable. And the other thing is, with a breath test, it's really hard to do cross-reactivity testing. First of all, the actual analyte isn't the virus or other viruses. It's volatiles, and so how do you get at cross-reactivity? And really the only way we could think of that's very doable and will allow developers like yourself to move forward is to test symptomatic people with a SARS test and also a respiratory panel test. Both should be authorized. There are some respiratory panels that have been authorized that have SARS on it, so it'd be one test. But you test, a sufficient number of symptomatic people. Somewhere around maybe 5 or 10%, maybe 10% of them are going to have SARS today of the symptomatic population. The rest of them are going to have some other respiratory disease, mostly viral, rhino, corona, non-novel corona, et cetera. So RSV, some flu, et cetera. So you should, without having to test that many patients, you should be able to see, and a good panel that would detect most of the non-SARS viruses that are infecting patients these days and causing symptoms. You wouldn't take that many patients that were symptomatic with this kind of comparison study to show if a breath test is specific for SARS or not. So that's our current thinking. If you send us an email at our templates email address for an update on our current recommendations and thinking for breath tests, we can provide you with some more information than we have in the past. Okay?
Yes, thank you.
Our next question is from Ling Ko. You may go ahead.
Hi, Tim. Thanks for taking our questions today. I also have a question about a smartphone-based reader test system. And I was just wondering, when asked to provide hardware requirements for a test system that includes a smartphone and a test, does the agency have a standardized list of requirement categories, for example, processor type, camera, et cetera, or is it sort of up to the test developer to provide that list of requirements, assuming they can then justify why those are relevant to the submission?
Yeah, we take an open-minded approach, Emily, to this. So we don't really know what the needs of your test are, right? You know it better than we do as far as the requirements on whatever the variables are when it comes to smartphones and cameras and image analysis. We also don't specify that you have to use a smartphone to read a test, right? But I'm trying to think if we've actually authorized a smartphone reader for SARS or anything. I don't remember that we have. So as with usual with something that's novel, and we're totally open to it, we learn a lot with the first developers who come to us with data and learn a lot about what our recommendations for others would be based on our actual data. It's really hard to we do have a research branch in our center who could look at something like this, but they aren't, to my knowledge, at the moment. They have so many interesting things to study. But one of those is actually digital health. So digital pathology, rather, which is an image scanner and analysis of tissue. So we do have a program there. And that's probably the most amenable thing that we understand relative to this. But there's a whole lot of variables when it comes into scanners of a rapid test, lateral flow test. From what is the color of the line, to the thickness of the line, to the lighting, to the background, and obviously different cameras or different software may impact that. So you're really the best one to inform us based on your examination of different smartphone options and your test what's going to work best and how you define that. And then we'll look at your validation when you define those parameters and determine if that is something we can authorize.
Okay, thank you. Is this something that you would recommend that we reach out directly for a more detailed conversation around, or can we just present the data as part of our submission and go from there? It never hurts to send us a pre-EUA and state your thinking and study design, because we may spot things that you may not think of, which would help you in your development and to prevent any hurdles later on where you might have to do some repeats or repeat testing. So especially novel, if we can take a quick look at it and spot anything that's obvious. But as I said, this is going to be new for us to know what are the variables around accurately reading a test over the smartphone, and what are the important questions for us to ask in the validation plan. Okay. Great. Thank you so much.
Our next question is from Raymond Boulay. You may go ahead.
Actually, some of the previous questioners have actually addressed my question, so thank you.
You're welcome.
And our next question is from Kay Taylor. You may go ahead.
Yeah, Tim, I just wondered if you had an update on the reference panel testing by the CDC for EUA authorized antigen tests. Have they started reaching out to antigen manufacturers to request samples of the product? Can you just give a general status on where that is?
Yeah, I think they're getting relatively near to starting, but the CDC is inundated with mission requests, and we are currently thinking of other options and looking into them. And we don't have what I would call a great option right now. For those developers that can utilize in their assays the various strains that have been deposited at BEI and either have been heat inactivated or perhaps more appropriately irradiated and inactivated through irradiation that may be more amenable to antigen tests. We're going to be reaching out. We have some preliminary data within the federal government that says that irradiated SARS can be accurately detected by antigen tests. So, but I don't know if that would be across the board for all antigen test developers or not, if their test is sensitive to irradiation of the virus or not, as far as reactivity and inclusivity testing goes. But if it is amenable to all or most, one option is that once new strains are deposited at BEI and having been inactivated by one or another method, those developers that don't have BSL-3 facilities can get that material and perform the testing. So we're just working through that. The downside, I'll just say to that, is it takes BEI a while to inactivate virus, and obviously they don't just irradiate it or heat denature it. Then they go ahead and test it to make sure that it's non-infectious. So it's not something that they can rush through and do. So we're hoping long term the inactivated virus material that BEI manufactures will be useful for this effort. So that's our current thinking as of today.
Okay, thank you.
Mm-hmm.
And our next question is from Franco Calderon. Sir, you may go ahead.
Hello, Tim and Toby. Thank you again. So my question is related to, I actually have two related questions, but one is related to the last question. So we actually have been able to find a BSL-3 facility to conduct the analytical studies, and they looked at the requirements on Part J of the antigen test, and we want to go home use, so we have to refer back to the other one. And they have informed us that based on the requirements there, that they're not going to be able to help us. So my question is, if they are able to, let's say, complete most of those analytical studies, would FDA be amenable to maybe allowing us to do the remainder post EUA? And that's one question. The other question is, I've noticed that to date, I believe there are 13 EUAs for antigen tests, and none of them are yet from a company that has an OEM based in China. And that is our case. So while we are able to gather up resources in China, OEMs that can potentially produce tens of millions of these tests per day, if we put 3-4 of them together, I wonder if you can give me a realistic feedback regarding one, the possibility of a Chinese OEM getting EUA, because I haven't seen one, and two, how difficult would it be to have 3 or 4 OEMs producing the same test for a test developer, provided that obviously is the same test?
So I'll answer your second question first. EUA authorization holders can expand manufacturing to new plants. They don't have to, and there's no FDA review of that. We expect you to, the performance of the test in the new manufacturing site matches the performance at the original manufacturing site, but that it's not something that under EUAs that we review and make an authorization decision around. As far as foreign OEMs, we treat everybody fairly. We make decisions based on data and the quality of the submission. It doesn't matter where it comes from. But we haven't authorized that many antigen tests compared to molecular tests. But very clearly, we've authorized molecular and serology tests from many countries of the world, including China. So it's not it's not any sort of global issue that I know. So what matters is the quality of the test and the quality of the submission. That's what matters to us. Okay.
Okay. And regarding the first question, do you? So by the way, my question was not whether FDA had a bias towards a particular nation. That was not, what I meant. If that was perhaps what you interpreted, my question was perhaps have you seen a pattern from companies that are submitting tests made in specifically antigen tests where the performance is not meeting FDA requirements, in this case for home use, 90% sensitivity. And so the other one, again, would FDA be amenable to, let's say, the BSL-3 is able to complete most of those requirements on Part J, the analytical part? Would FDA be amenable to a post, to letting us do that post EUA?
We've been flexible when it comes to authorizations and what can go into a post-authorization study to fill out an application. We want to have a good sense of the performance of the assay, and we very clearly allowed on a case-by-case basis, depending on the benefit-risk calculation on the submission, what we have, to allow post-market studies to fill it out. I would, as far as manufacturers of antigen tests go, here's the maybe not well-kept secret, or maybe it is a secret, that it's a lot harder to develop a good antigen test than it is to develop a good molecular test. Now, I've developed both, and I can tell you it's a lot easier to develop a good molecular test than a good antigen test.
Okay.
And it's also much easier to manufacture molecular tests than antigen tests, and I've done both. So that's probably more of a factor here. Lateral flow tests and antigen, there's serology and antigen tests. I'll let you know. I mean, we're declining 2/3 of serology tests that are coming, lateral flow serology tests based on NCI data. 2/3. We're only authorizing 1/3 that have had tests performed at NCI and reviewed by the FDA. I think that's the nature of the difficulty in making really super high quality high enough for the U.S. market in the lateral flow business. And it probably is the same in antigens. I know we've denied some antigen tests. I haven't done the numbers as far as the ratio goes, but it wouldn't surprise me at all if the ratio was very similar. That is 1/3 authorizable and 2/3 not, based on data.
All right. Thank you very much.
And I show no additional questions at this time, but again, if you would like to ask a question, please press star 1.
Operator, I will give it a few minutes or a few moments to see if we have any more callers coming into the queue.
One more coming in right now. One moment.
Okay, thank you.
Our next caller is Christopher Benson. You may go ahead.
Hi, Tim. I had a quick question about UDI requirements for an EUA test. I can't find it anywhere in the recommendations. I'm assuming the UDI G10 requirements for Class 2 and higher tests are not applicable right now for an EUA.
I believe they are not applicable. Toby, do you know?
I believe that is the case. It would probably be best if you can send that question in to the mailbox so that we can confirm, but I'm pretty sure that they are not applicable.
Yeah, it'd be nice if it was somewhere locatable for people who can find it easier than, sending in emails. But thanks for the advice.
You're exactly right, Chris. Yep. And I was just going to ask Toby to take a note. Let's find that out and provide that definitive answer on the next town hall call, and consider whether or not we put it on the frequently asked questions page.
Yeah, that would be great. Thanks, Tim.
Appreciate the feedback. Thank you.
Our next question is from Gloria. You may go ahead.
Hello. Yes. I have a question on the reporting. So we are developing an antigen test, and we are willing to provide it with and mobile app for guiding the user through the test and for reporting the result could also be an option. If we're reading the HHS specifications for COVID-19 data reporting, it's very oriented towards point-of-care testing. So we are wondering if there is some guidance in case of reporting for an at-home test, what would be needed on personal data and on data of the user. And also, do we have to encounter a mechanism to predict and to say something about the prevalence in the region the consumer is behaving? So you could give an indication on the PPV and the NPV, so it's more trustworthy, the results.
I'm not sure that I caught the last part of your question. The first part I caught was you're developing an app to guide the person through the testing and to report the results. That you aren't using the smartphone to actually read the results, but maybe you are.
No, it's read out on the reader.
Reporting, it's guiding and reading. It's guiding them through and reporting the results. And then you want to know what are the point of care or home recommendations for reporting and how you do it and how you ensure confidentiality and you're mindful of HIPAA requirements. And those are all really important questions. And the facts are that we are still struggling with how to do that reporting well. Even if you have an app to report, to connecting it to the database and getting authorizations from all of the states and the federal government to be able to do that and connect it all up and report results. That is a mission in process. And we're fortunate to have our office, the office I lead as a chief medical officer, who's an expert in this, Dr. Sarah Brenner, who's actually on loan right now to HHS to work primarily on this reporting question. And it's I know that there's websites and there's documents that are available, but I don't have them at my fingertips. Sarah would. It's probably something, Toby and Irene, that we want to add to our slide deck that we provide at the town hall, some materials around reporting, and since we've had a number of questions about reporting, and those links to important documents that can guide developers. But we're very interested in working with you. So if you send us an email to the templates email address and you ask for that to be forwarded to Dr. Sarah Brenner, Brenner, Sarah will get back to you with as much as she can tell you about the process now.
Okay. My second part was then on the prevalence. Could it be, it was already discussed earlier, that RX, the prescription, as a mitigation of risk, does FDA also consider a prediction of the prevalence of COVID-19 and the region where the app is installed and where the user is doing the test as a mitigation of risk? If it is a low prevalence region, then our result might be less true positive. That kind of mitigation, is that open for discussion?
It's an interesting question. Certainly, in a low prevalence population, your PPV is going to go down. Your positive predictive value is going to go down, but your NPV is going to go up. But we've heard that false positives with antigen tests, especially in point of care, congregate settings, where then they move a patient based on their test results into a COVID ward, can expose someone who doesn't really have COVID to SARS and developing COVID. So it's a complex thing. The other thing is, and obviously if your prevalence is higher, then it's kind of the reverse. But one of the challenges with that is you'd have to have, first of all, even to consider it, you'd have to have really good prevalence data and up-to-minute prevalence data, which we don't have. And then you'd have to have an adaptable system that had some geolocator that says, well, you're in this zip code and this is the prevalence, and so this is the number to use, for your mitigation. So it's an interesting idea. I don't want to give you any more definitive. I don't want to give you any definitive answer on it. We're always open to new ideas.
Okay.
So if you want to propose this in a pre-EUA or an EUA as a mitigation, please do. I can't guarantee the results, but like I said, this town hall is to help educate folks about our current thinking, but we learn just as much from developers, as you do from us. So, thanks for asking a creative question.
Okay, thank you.
Our next question is from Mac Clement. Your line is open.
Hi there. Thanks for taking the call. This is a little bit off base, but I work for a company where we do both medical distribution and then we're also doing development as well. So we have a biotech arm. We have our own med techs, et cetera. We've had a few positive tests within our own organization and are going to set up, we hope by the end of this week, some surveillance testing. There shouldn't be any issue with that, should there? Using our own tests, using the tests, not tests that are not developed yet, but tests that we're already distributing, antigen tests. Does that make sense?
So, it's a bit complicated. So, first of all, the FDA has tried to provide some clarity on this, on our FAQs with regard to what is diagnostic testing, what is screening, asymptomatic screening testing. And what is surveillance testing. And as clearly said, that for COVID-19 at least, the FDA is not going to be regulating surveillance testing. So, but one of the challenges, what you mentioned with the antigen test, is it's a single use typically for a single patient. And it's hard to do purely surveillance testing when you have one patient and one result, and it's right there, and the patient sees the result or is the test itself. So that isn't, when a patient gets a result back or an employee gets a result back that tells them that they're positive or negative, that is not what we would typically think of as surveillance testing.
Okay.
Surveillance testing is population-based. Typically, it uses pooling. And then if you have a positive pool, at least the FDA current thinking on this is that if it's a positive pool, you can say to that pool of people, go get a CLIA lab test and get a result. And to the negative people, you can't say you're negative, but you don't direct them to get it. Then you don't direct them to get a CLIA test.
Okay, thanks. Appreciate it.
And our next question is from Rainer Ziernan. You may go ahead.
Hi, thank you for taking my call. I have a question regarding requirements of usability study for molecular point of care claims. The July 28 template doesn't really specifically outline the requirements, and I'm aware of the other template dated July 29, which refers to human usability studies for non-prescription OTC tests or for prescription-only tests. So do I understand correctly, for a point-of-care molecular test, we should follow that guidance and include 30 participants in that human usability study? Or if not, could you just quickly give me some advice on what is the extended scope that's required for the point-of-care claim? Thank you.
Okay, so you have okay, hold on. I just want to make sure I understand. You have a point-of-care test. It's molecular test?
Correct, yes.
Okay. And you want to know, this is point of care. There's not home. It's just point of care test. You want to know how you do the user in a ClearWave setting testing? Because I don't remember that we had 30. We have 30 positives for the point of care clinical study validation. And then we do want to see, it would be great to see more than one clinic, although I don't think that's a formal requirement. But we do like to see 5 to 6 different people, if I remember correctly from the template, perform the test, and not just one person in one clinic performing the test. If you move into the home setting, either OTC or RX, then we ask for clear user studies, and the numbers are bigger for that. I don't really have time to pull up the template for point of care molecular, but I thought it was fairly clear. If you have any clarifying questions about what's in the template, what you need to do, it's best handled through our template email address. But I think I roughly covered accurately. Toby can probably tell me if I'm wrong or not. What the user studies are for point of care tests.
Yeah. Tim, can I just...
But it's, there's discussion about the operators, the site and test users, and how to evaluate that as part of the clinical evaluation.
Yeah, I guess maybe I wasn't entirely clear. I'm fairly clear on the clinical study and what we do there, but I was wondering whether we are required to do an additional usability study that will be part of the submission beyond the clinical study.
Not for point of care.
No, I believe we built that into the discussion, the clinical study, so that it would streamline things for you.
Okay. Thank you very much. I appreciate it.
Yep.
And this concludes our question and answer session. I will now turn the call back to Irina here.
Thank you so much. This is Irina here. We appreciate your participation and thoughtful questions during today's town